everyone, my name is Yo-Yo. I'm a PhD student from the CS department of the City University of New York. Today, I'd like to introduce this method called MAPLE, which stands for Meta-Model Agnostic Pseudo-Label Learning for Understudied Out-of-Distribution Molecular Interactions. And I was inspired by their two challenges. The first one is data insufficiency, which is even more than the typical machine learning task because in the biological domains, there are more technical limitations prevent us to have amount of label data needed to develop the machine learning methods. There are few examples to illustrate this idea. There are more than 90% of protein families that we do not have their known ligands yet. And out of more than 3,000 brain cell types, there are only 100 labeled. Not to mention the chemical space that's supposed to contain more than 10 to the power of 60. However, there are only 10 to the power of 6 are known. Besides that, there are also a lot of distribution shifts that further challenge us. This is also what we call OOD, out of distribution problem. And this happens when you have training and validation from one distribution, however, the testing from another distribution. This will normally lead to the decrease of the performance versus the performance that you had when you testing on the in distribution samples. But this is very important and closer to the real life application because oftentimes you apply your developed model on the scenarios that or on the tasks that the model never performed before. So we want to develop those generative AI model to not only generalize well on the in distribution samples but also on the out of distribution samples. So we couldn't help wondering if we can really leverage those unlabeled data space to boost the existing model performance and also address those out-of-distribution problems. The answer is this method called MAPLE. And here are the overview of the model architecture. So given the pre-trained teacher model, we copied this architecture to have our student model. And the student model will learn from the pseudo label generated by the teacher model. And up to here are the traditional teacher-student model. And then we introduce this idea of meta-learning, which happens here when the model is queried by the label data to assess the performance of the updated student model. That state of student model was only learned from the pseudo label. But once you introduce this meta learning, and this meta update will further give this feedback to the teacher model for the model to learn that how well or how bad the updated student learned from the previous pseudo label. And then the teacher model can adapt to the feedback to upgrade the pseudo labels in the next iteration. And this procedure happens iteratively until the student model converges well on the validation set. And we tested our method on 3 scenarios. The first one is OOD drug target interaction. And the first column stands for the ROC AUC curve, and the second one is the PRUC curve. And each row stands for one state-of-art machine learning methods. And because MAPLE doesn't perform alone, so we end this MAPLE training on top of those 4 methods to see if the model can bring significant performance gain. And the results showed our model constantly bring these gains on top of these 4 state of methods. As you can see, this orange, which is our method, is always better than the blue one. And we also want to know if our model can, draw these 2 distribution together. And the first one of this U map shows the original space of 2 distribution, which is randomly located. However, the second one is after the baseline. You can see there is a slightly better alignment going on until the third one, which is after our maple training, which perfectly bring these two distribution closer. And the second testing scenario is a similar idea, but on the hidden human metabolite protein interaction. And the third one is the results of the interaction between TIMO and the human GPCR. And the way we did this was we firstly picked the top 7 predictive genes, and out of them, we picked the one with the strongest antagonist activity, which was the CXCR4, to perform the protein ligand binding docking. And this is the 3D binding pose, which suggests that the TIMO was able to fit into the structure of the CXCR4. And the lower picture suggests the mechanism behind these 5 genes. So there are 2 genes that there is just no assays available to test them. And it's known that the CXCR4 is responsible for those like PI3K and this RAF MEK pathways. So, and which leads to the further phenotypic effects. And this also supports our hypothesis. Yeah, that's the end of my method. And lastly, I want to thank our dearest professor and all our lab members. Thank you.